Up next, please welcome our next group of speakers to the stage to discuss why do women have a higher burden of disease. Moderating this session is Dr. Jennifer Garrison, a pioneering neurobiologist, entrepreneur, and women's health advocate whose research bridges metabolism, ovarian aging, and brain health. Dr. Garrison holds faculty appointments at UCSF and USC. Let's give it up for our next panelists. Thank you.
I am delighted to be here. So we're switching gears a little bit. We're going to talk about some science, and we're here to talk about sex differences. Now, male and female bodies are different. You don't need to be a scientist or a doctor to understand that. You can see it with your eyes. But those differences extend deeper to the molecular level, to the cellular level. And there are diseases and health conditions that manifest differently between men and women, obviously. Things like heart disease, very different in terms of how those symptoms manifest. Then there are conditions that are specific to one sex or the other, like, for example, prostate cancer or endometriosis. And, at the end of the day, we know a lot more about how male bodies work. We're not here to rehash the problems. I think everyone in this room can agree and understands that there has been societal taboos, there has been a massive underfunding and a massive lack of investment into understanding female bodies. And as we move forward and think about how to solve these problems, to think about healthy aging in women, there are a lot of things that we want to consider. So one seed that I'll plant right now, just as you're going to listen to these amazing scientists talk about their work, is women live longer than men on average, and yet we spend longer, if we're lucky enough to live to old age, females will spend a significantly longer period of time in poor health at the end of their lives. So another way to say that is that they have a shorter health span at baseline. So why is that? And we'll hear about some of that from these amazing scientists. The other thing that I wanted to say as I'm introducing them is as we're thinking about healthy aging and how to extend health span in women, we really want to focus on the systems that are specific to females, but understanding those sex differences and it extends beyond just improving women's lives and improving women's health spans. When we miss differences between the sexes, it actually has a dramatic impact on understanding male bodies as well. So if we miss sex differences, we're essentially missing breakthroughs that could impact men as well as women. So I think that's something really important to keep in mind. Without further ado, I would love to introduce our first speaker. So we're going to have them give short talks, and then we'll sit together, have a few questions, and then get input from the audience. So I'm delighted to introduce Dr. Dina Dubal. She is a professor of neurology and an endowed chair in aging and neurodegenerative disease at UCSF. She's a physician scientist, which means she's one of those rare unicorns who both sees patients and also does research in a lab. And she studies brain resilience and longevity. So welcome, Dina.
Thank you. Delighted to be here. Thank you to the Cohen Foundation for highlighting this critical topic of women's health. I'd like to start with some personal history, if I can share some. This is probably the origin of my curiosity about women's health and now my commitment to women's health. My great-grandparents, who lived in a very small village in West India during the time of British rule. And I did meet my great-grandmother there on the left. She was absolutely remarkable, uneducated, survived malaria, smallpox, probably tuberculosis, mourned 4 of her 8 children, and yet there was some resilience there. She lived to be 90 years old, despite these stresses of the environment, and she was extremely sharp up until the end. Totally remarkable. And my great-grandfather died in his 40s of infection, and I was really struck by this difference in lifespan and grew to understand over the years that this was not specific to my family, but in fact, this is universal all over the world, that women will live longer than men. This is data collected from the World Health Organization, and you can see that in every society that records mortality, from Somalia to India, the United States, Korea, Japan, women will live longer than men. This is true in neurodegenerative diseases like Alzheimer's, ALS. Women live longer than men. We see this across the animal kingdom. In many species, the females live longer than the males. When we even look at brain cells in a dish, those XX cells will survive longer than the XY cells. So there's something deeply biological here. And what does this mean for women's health? It means that there is a biology of resilience that is waiting to be studied and uncovered. Now, living longer, as we heard from Jennifer, there is a resilience, and then there's also vulnerability, and those two things come hand in hand. There may be more chronic diseases, for example, osteoporosis, muscle wasting, increased risk for Alzheimer's disease, for example. But we have been studying the resilience aspect of female longevity. And what we're finding from data in humans and in mice is that there is a remarkable resilience in the aging brain. Despite the declines that happen, females are resilient. And we know from early studies and recent studies that in typical brain aging, in the absence of dementia, women show less cognitive decline compared to men. What about Alzheimer's disease? In early Alzheimer's disease, there's an increased baseline function in women in terms of their cognition, particularly in verbal memory, compared to men. And then they have more pathology, more tau, more amyloid beta in their brains, but they're able to hold on longer. They're bearing a greater burden, but they're hanging on longer. That is resilience. And then finally, when Alzheimer's really is in its throes, women will decline faster, and that's the vulnerability part. Declining faster, vulnerability, resilience, higher baseline functions, and hanging on longer with more toxins in the brain. So, well, when we think about what this means, what we can learn from it, we turn our attention in the laboratory from observation to the how, the mechanism. So what is the biology behind the resilience is what our focus is. And to do that, I lead an incredible team of physicians and scientists together who use powerful tools using mouse models to understand the how of the resilience, to understand whether it's hormones that are contributing, whether it's sex chromosomes that are contributing, whether if it's sex chromosomes, is it because of the X or is it the Y? If it's the X, is it because of mom's X or dad's X? Is it the active X, which I'll tell you about, or the inactive X? So there's a really powerful armamentarium of models that we use to really decipher and discern the causes of female resilience. And who cares? Well, we care because this can lead to new medicines. If we can figure out the how of resilience, we want to turn those molecules into new medicines to amplify resilience in the female brain as well as the male brain. In many, many of these studies we've done over the past decade, our data points to the second X chromosome in females. So we have found that in mice, that second X will contribute to living longer in females, as well as to resilience in the brain, but how that's happening is unclear. In development, when we were literally a few days old, in a female cell, there are two X's, and one of them shuts down. So one of them is active, and one of them is inactive in every cell. That's random X inactivation. So what we found in a major discovery earlier this year is that aging wakes up that silent X in women. And this was totally remarkable because it has long been thought that once that silent X shuts down when we're embryos, that was permanent. But we have, again, a new finding that aging will wake up that sleeping X. And what that means for a brain cell, is in a female brain cell, is that there is more of the X being expressed. We have the chance now to ask, well, what about other diseases? Does Alzheimer's wake up that second X? And then what does that mean for resilience or vulnerability? And can there be new medicines that we can develop from genes on the X chromosome? So much to come there. I wanted to show you a key study in that we took one of those X genes on that sleeping X that wakes up. We just took one of those X factors and we boosted an old mouse brain by increasing that factor. And what we found by increasing that gene from the silent X is that it improved cognition in an old female mouse brain, an XX brain, as well as in an XY brain too. So boosting up this X gene from the silent X improved brain function in an old female and male brain. And this leads us to two paradigm-shifting conclusions. One is that the X chromosome may be a treasure trove of therapies that we just haven't discovered yet. When an X is mutated, for example, there's intellectual disability, and the X is enriched for cognition genes, maybe a treasure trove. And secondly, when we discover fundamental biology from a female brain and female biology, that has the power to affect both female brains and male brains. I leave you with that thought and acknowledge the people who did the work and my funding sources. I look forward to a discussion.
Thank you, Dina. That was really interesting. I'm delighted to introduce Dr. Amy Proal. She's a microbiologist, president and CEO of PolyBio Research Foundation, and the scientific director of the Cohen Center for Recovery from Complex and Chronic Illness at Mount Sinai. Welcome.
Thank you. Okay. All right. Great. So today I want to talk about why women have a higher burden of chronic disease, but bringing in the infections that all of us acquire into our bodies over the course of life into that equation. So again, all of us, sometimes starting in the womb, inherited from our mothers, and then across the course of our lifetimes, through close contact, through other exposures, through mosquito bites, through spider bites, through all kinds of vectors, acquire viral, bacterial, parasite pathogens into our bodies over time. And these infectious agents do not often just sit there doing nothing. One of the things is that right now, if you look at the study of chronic disease, so you're looking at multiple sclerosis, you're looking at schizophrenia, you're looking at the other autoimmune conditions like lupus. People are putting these infections right in the center of the activity, for example, of how these diseases happen. So it's important to understand that this trend is happening. Now, we know then that especially, let's say, with the autoimmune conditions, many of those conditions impact women more than men. And why? Could there be some connection that involves the infections in that? So one of the conditions or types of conditions that we study in this large picture of infectious contributions to chronic disease are literally what we call infection-associated chronic conditions. So someone gets an infection like SARS-CoV-2 or like Borrelia, the Lyme pathogen, and then they do not recover. Instead, they go on to develop extremely debilitating symptoms in many cases that can last for a lifetime. Now, a central thing that our groups are studying in patients with these conditions is a really straightforward possibility for why at least some of these patients are sick. And that is really that they may not fully clear the infection that they get, and that the infection, let's say it's long COVID, and it's the SARS-CoV-2 virus, a small amount of that virus persists in their bodies in what we call a reservoir, where it may be able to drive inflammation and other problems across time. And there are many groups in the long COVID space now showing that SARS-CoV-2 is capable of long-term persistence, for example, in the gut, in the tissue of the gut, even, for example, up to 2 years, even after infection. So a thought there then is that there are certain sites in the body that might permit or can permit this chronic infection from happening more often. One is the gut, potentially, because it's a tolerogenic environment. And what I mean by that is all of us have microbiome ecosystems in our gut, trillions of interacting bacteria, fungi, all these ecosystems there that help us digest food and do all kinds of other functions. So if the gut, every time a new organism or a foreign invader came in, had a profound immune response, that would be very difficult in the context of the fact that it regularly has to contend with these foreign agents as part of the natural microbiome, right? So it doesn't do that. It has a tolerogenic environment where it tends to have an immune environment that's more okay with infection, which is possibly why the SARS-CoV-2 virus can persist in some people in a reservoir there a little bit more easily, okay? Another site that's tolerogenic in the human body in women is the female reproductive tract. And that's because it has to accommodate sperm, which are foreign. Also, if there's a fetus that's, implanted during pregnancy, half the genetic material from that fetus is from the father, so it's foreign, partially the fetus. So the atmosphere in the reproductive system is also tolerogenic, which means that the female reproductive tract may also be an optimal site for some of these pathogens when they hide out and persist in a reservoir. And indeed, there are 2 groups now we're working with collecting tissue from the female reproductive system via a biopsy procedure from women with long COVID. And early data shows that there could be a persistence of the SARS-CoV-2 virus in that site. Okay. Now, another major trend in this space of women and chronic disease and incidence of women being higher than men in some conditions involves the nuclear receptors. The nuclear receptors are hubs. They're major receptors that are hubs between the endocrine system and the immune system. So what happens is, for example, the estrogen receptor is one of these. It controls estrogen signaling, hormonal signaling, but under control of that same receptor are key parts of the immune system. In fact, even key parts of the innate immune system in some cases, that's just so important in recognizing and dealing with infectious threats over time. So this is a thing that multiple pathogens have evolved to do. This is just an example with SARS-CoV-2, but there's data on other viruses, other bacteria. They create proteins that can directly dock into these nuclear receptors, like the estrogen receptor, and modulate the activity of the receptor, likely in a way that throws off estrogen signaling, but then has a flow-on effect of potentially dysregulating many of the components of the immune response also under control of this receptor, which can lead to a situation where there's immune dysregulation that might make a woman less likely to be able to clear or manage an infection they have, and this is kind of like a feed-forward loop of issues with infection, right? So this is a really important understanding that when we're looking at hormonal signaling in the human body, we have to continually say, how are the pathogens that can get into us and their proteins modulating that signaling? We can't leave that out of the picture. And then this is just the last thing. Overall, there's evidence that infections can be found often in female reproductive tissue or even endometriosis tissue if we just check. And so few people do this. So for example, part of what Polybio Research Foundation, the nonprofit I lead, does is convince surgeons to not throw out the tissue samples that they obtain from women or other people when they perform surgery. So we have a study where women are undergoing surgery for endometriosis, but we save that tissue and we stabilize it. And there are teams looking for different viral bacterial pathogens in that tissue. That study is underway. This is a team already, though, that what they did is, this is a case where they obtained tissue from a hysterectomy from a woman. And when they looked at this reproductive tissue, they found Babesia oticoli, which is a parasite carried by ticks that is actually more frequently infecting people than I think is recognized in the tissue from this woman. And it was a very high amount of Babesia in the tissue. So it's, again, proof of concept. There can be organisms in these tissues that we are just not accounting for often. And last, this is a paper by Mickey Tall's group and Linda Griffith at MIT. They did a mouse model. They infected mice with Borrelia, the Lyme pathogen. Then they looked over the course of 15 months. It infected the female reproductive tract. It caused ovarian cysts. There was endometrial hyperplasia. There was thickening of the vaginal wall. This went on over 15 months in these mice where the organism did not clear and was directly associated with these issues. So, and the older mice in the study had more problems with this than the younger mice. So again, this is a frontier. We have to consider these infections that many of us get and are exposed to. They may very well be one of the reasons why women suffer from more disease than men and affecting the chronic conditions overall. Thank you.
I hope you're all learning something new here. This is really exciting. And last but not least at all is Dr. Yushin Se. She's coming to us from Columbia University, where she is professor of reproductive sciences, genetics, and development. And she also leads the Center for Reproductive Aging in Women. And she is a human geneticist who studies age-related diseases and resilience.
Oh, thank you so much. That's a very kind introduction, Jennifer. Thank you to the Cohen Foundation. I'm very happy to be here. It's an exciting event. My lab at Columbia has been addressing one major unaddressed question in the field. That's why ovary undergoes aging so much faster than the rest of the body. So we think this question is really deeply connected to a bigger issue. That's how women suffer from higher burden of disease. That's the topic of this session. So we want to understand what drives this accelerated aging in the ovary. Sorry, forgot to show this. And more importantly, how we can slow it down or even stop this acceleration, because we think it has enormous impact on overall health in women. So as you will see, we are one step closer to this goal, because by targeting these root causes of ovarian aging, we want to help women live not just longer, but healthier, as you see in this vibrant centenarian women, right? So aging, when you're talking about aging and also ovarian aging, there are many forms. I mean, it's just beautifully captured by this painting by Malevich, that no single woman aged the same way. It's reflecting this enormous heterogeneity or variability in aging forms. So we want to understand where this variability comes from. I mean, in previous session, we discussed timing of menopause. And this lady undergo menopause in her 40s. premature age, ovary insufficiency, you're talking about in 30s, where some women undergo aging or menopause in late 50s and early 60s. So how do you explain this? We believe that differences in our DNA contribute to the variability. So what we are trying to do in the lab is to identify genes that drive this premature aging in the ovary. And the way we go by is to take ovaries from women, different age groups or across lifespan, and try to understand what happens during aging processes in the ovary, at least at the molecular level. And once we understand these molecular changes from the profiling, we are going to integrate with the human genetic data. So I'm talking about human genetic data from hundreds of thousands of women. So by incorporating this information computationally, we can really predict who are the genes that may contribute to this aging rate in the ovary. And once we have those lists, then we can actually bring those lists to the bench in the lab using ovarian cell types, using these wonderful cutting-edge tools like genome editing, very high throughput, massively parallel way to validate if our prediction is correct. And once we identify those, then we understand how this gene contribute to modulating the rate of aging in the ovary. So what have we discovered thus far? So this is a cloud of genes that, at least in the lab, we have shown that contribute to aging rate in the ovary. You can see some genes are bigger than others, so gene names, which represent more impact. But you may not know, but we know that many of these genes, we have no idea how they function in the body. So I see several people from the lab, and they are working really tirelessly to understand how these genetic drivers of ovarian aging contribute to this premature aging in the ovary. So at the end of the day, what we want to do is that if we identify these beneficial genes that mean delaying aging in the ovary, we want to mimic those by using maybe nutritional pharmacological interventions, or if we identify deleterious genes that may further accelerate aging in the ovary, we want to find a way to counteract this deleterious nature of the genes. What about the molecular signatures? So when we start this project, we really had no clear hypothesis because, as Jennifer said, really nothing much has been done. We know very little about how female body works, let alone physiology, or at the molecular level. So we could suspect that maybe ovary has ovary-specific genes and pathways, but we were blown away to see that the usual fundamental processes of aging that you see in other body parts, brain, breast, lung, heart, you name it, occurs in the ovary, but much, much faster in such a compressed way, right? So for example, here I show this mTOR signaling pathway, which is really a central regulator of aging, right? And its inhibitor, called rapamycin, extends lifespan and health span in all preclinical models that have been tested thus far. So rapamycin has been known as the most robust, what they call geroprotectors, right? So our results showing that this mTOR signaling is extremely high activated in all cell types in the ovary. That's you indicate in these little boxes in different cell types, not just follicular cell type, but all cell types in the ovary, suggests to us that, oh wow, ovary can be a zero therapeutic target of rapamycin. Okay, so of course the big impact comes from to test this hypothesis, right? Does rapamycin truly delay aging in the ovary? So we have started clinical trials. It's called the Vibrant study, validating benefits of rapamycin for reproductive aging treatment. This is one of the very first its kind. We are targeting women in their 30s to early 40s to see rapamycin, who's known to delay aging in the body, can delay aging in the ovary, premature ages, right? And see if it improves reproductive lifespan, thereby promoting overall health in later life, in women's life. Okay, with that, I'll stop here. Thank you for your attention.
Wonderful. That was amazing. And hearing about things from so many different directions, chromosomal biology, infectious disease and immunology, and then ovarian aging, I can see the through lines between all of the talks that you gave. But I'm curious, listening to each other's talks, were you surprised by some of the things that are similar here?
Absolutely. We're focused on the brain and brain resilience, but this is not a zero-sum game. The brain is going to be interacting with the ovaries, with the immune system, and there will be convergence, there will be divergence. And so, like really digging into the biology, the fundamentals of each of these axes is going to be really important for the future of women's health. I wondered, for example, whether, if you slow down ovarian aging, might that actually thwart Alzheimer's disease? I also wonder a lot if we can slow down brain aging, whether the whole body aging would be slowed down. Maybe women would be less vulnerable to infections. So there's quite a synergy that will be important for women's health.
Sure. One of the things I found most interesting is your clinical trial of the low-dose rapamycin for ovarian health. We're running at the Cohen Center for Recovery from Complex Chronic Illness, which is here at Mount Sinai. It's a clinic where we treat patients with long COVID and chronic Lyme disease and related conditions. We're running a trial of low-dose rapamycin, I think almost in the same dose as your trial for long COVID. And I guess you could ask, why would this drug in the same, it's a once weekly low dose, maybe also help patients with long COVID? Part of that comes from the fact that there's data showing that in a low dose, once a week taken, rapamycin might actually support parts of the immune response, potentially in a way that allows a person to better control infection. So there are actually 2 trials, this is Matt Kaberlin's group and a couple other people, where in healthy older adults who are taking this low-dose rapamycin, they measure interferon signaling, and interferons are really key proteins that help the body manage viral infection especially, but really most infections. And then also they measure T-cell exhaustion. And if you struggle with an infection over time and you don't totally clear it, your T-cells can become more exhausted because they're immune cells that keep recognizing the infection, trying to clear it, but they're failing, so they literally become exhausted. So at the end of these trials with the low-dose rapamycin, the older adult subjects had increased, the interferon signaling improved. It was higher at the end of the trial, and one of them also showed that T-cell exhaustion was mitigated, suggesting that the low-dose rapamycin is improving those aspects of immune function, so maybe it can control infection then. One of the reasons why this might happen is because most pathogens have to hijack our mitochondria in order to replicate or do anything. So for example, if you're a virus, you're really what's called an obligate intracellular parasite. You cannot make a new copy of yourself. You cannot replicate. You can't do anything without pulling the substrates you need to replicate out of our own mitochondria. And every time that they do that, they throw off the metabolism of they throw off the mitochondria. And mTOR is one of the main pathways that so many pathogens dysregulate as part of doing that, that there are literally entire papers on mechanisms by which viruses and other pathogens hijack the mTOR pathway. So a drug like low-dose rapamycin that might impact what might happen, and it's just a hypothesis, is that perhaps low-dose rapamycin gets in the way of this hijacking of the mTOR pathway by lots of different viruses and bacteria by stabilizing mTOR signaling, preventing some of that hijacking from happening, which might allow the immune system to come up a little bit, improve over time in people who are taking it. And we'll see. So we're testing some of those hypotheses in our clinical trial, but long story short, in this huge overall picture, there could be some infectious components of why ovaries are aging, literally infections that might either directly infect or impact the ovaries. And if low-dose rapamycin can better help control those infections or just support immunity in people, that could be a synergy of why people might be benefiting.
Well, I think that's a very interesting point, and opposite can be also true because at least in preclinical models, the effect of rapamycin in extending, lifespan, protecting against age-related diseases, more pronounced in female compared to male. So we like to believe that because ovary is so screamingly high, show the pattern of mTOR activation, by dampening it with rapamycin, it's going to benefit them more. That's why female reap the benefit more. So going back to Dina, your comments about Alzheimer's disease, yeah, there are large body of literature showing that the timing of menopause is correlated with the risk of neurodegenerative diseases, cardiovascular disease, metabolic syndrome. So it's really critical. It's not just, so Jennifer really has set the stage about how important this reproductive aging in women and menopause. We heard from Paula this previous session that it really came together in 2023, but the narrative that we have is really very critical, right? So now everybody recognizes this is such an important field that we've been ignored for so long. So let's make difference now.
Let's do. Yeah, for those of you who don't know what she's talking about, we think about ovaries like a second brain, in the sense that they really are talking to almost every tissue in the female body. And when ovaries aren't working properly, or when they stop working the way they were before at and after menopause, that really impacts almost every system in a female body. And so understanding at a fundamental level how ovaries work at every age is really like the basis for how we move forward to really translate healthy aging in females. And we need that basic science, that knowledge, to understand how to prolong health span in women.
Jennifer, I wanted to add that I was also thinking as I'm listening to your talks and the bigger picture that just after we published that aging awakens the silent X in the brain of female mice, that another group a couple of months later published that aging wakes up that silent X chromosome all over the body. So it's not just happening in the brain. And again, to really understand the fundamentals of what does infection do to that silent X? What does aging do to the silent X in the ovary? And what does that mean for women's health? And most importantly, are there new medicines that we can develop from that knowledge for women's health and get it to humans for human trials? We'll get to this, but there's so much hope and there's so much potential to improve women's health. So much to do.
Yeah, absolutely. I'm going to ask a few more questions just to seed the field, but I want to leave enough time for the audience to ask you all questions too. Yushin, you often say in your talks, you refer to data that shows that brothers of women who go through menopause later tend to live longer. So we know there's an association between age at natural menopause and overall lifespan in females, but that also extends to male brothers. So are there genetic components you think there that are important for both males and females?
Oh, thank you for that question. Absolutely. So that's the reason why we are focusing on DNA. So there's a strong genetic components, right? So if you have family history of early menopause, your chances of having early menopause can go up to like 6 times. And interestingly, there, as Jennifer said, brothers of women with later menopause have pro-longevity benefit compared to brothers of women with earlier menopause. And when we did the genetic analysis, we were really looking for, can we find those genes that may influence ovarian aging as well as whole body aging through this, sex differences, right? And then we found such a gene which is correlated with later menopause in women. And then we showed that in the lab that it really has causal impact, right? It's not just association. But surprising thing was that effect was found in not just in the ovary, but all human tissues that have been profiled from brain all the way down to heart and even testes. Actually, the effect in the ovary was like in the middle, but the effect on the testes was higher, and the largest effect was found in muscle. And interesting, this gene is involved in really fundamental processes of aging. It corrects, errors in our DNA, genome stability mechanism. It's very critical pathways, right? So I think there's a clear link, right? So what we learn from menopause timing and female physiology obviously can be applied to male as well.
I think it's fair to say that the female body at this stage is like a puzzle that we haven't yet solved. And you can't expect to see the whole picture if you're only solving half of the puzzle. So this is really what we have in front of us, is to think hard about how to facilitate the critical fundamental research that's happening around female physiology and how to really promote that. I want to open it up to audience questions. Right here.
Hi, I'm Judy Regensteiner from the University of Colorado, and a lot of my work surrounds the area of sex differences. When I hear the basic findings that you've beautifully told us about today, I still come back to the clinical issue that there are no sex-specific clinical guidelines currently for women on such basic variables as blood pressure or statins. And I say we need to use our knowledge and information to get those guidelines. Unfortunately, a lot of those studies in blood pressure, for instance, were done a long time ago. Nobody's going to repeat them probably. So I've been working with AI experts to try to see if we can get data enough to create, something in that space. But that's, to me, where it all ends up. Women are still not getting evidence-based care. And there's a lot of evidence that there are differences in men and women, as we've all seen, and there's no question about that. So how do we get our work to focus on creating those guidelines so that women get evidence-based care?
I guess I would say that that's a space where the professional societies that put out the guidelines for clinical care should be playing a great role. I was just last week at the Menopause Society meeting, and I think in the last few years they've done a great job of increasing the amount of information they're putting out there. Now, I'm not saying that that's it's 100% at this point, but it's moving in the right direction. And I don't know, Dina, if you wanted to say anything since you are a practicing physician.
Yeah. Well, all of this is important because the fundamental science can be translated to humans. And so, if we can really invest heavily in the discovery, the fundamental biology, then we can know. We can have new therapies and know where the sex differences are and know the precise areas that can be targeted for both personalized health for women and for men. So for me, yes, there is so much to be done on the clinical realm, and there's so much more to be done to move into the clinical realm, beginning with the fundamental science.
Maybe right here.
So, Hadeen Jaffe from Harvard Medical School. Thank you all for the outstanding work you're doing. So for those of us who've been working in this menopause field for 2 or 3 decades, one of the key questions is to what extent the ovarian aging is contributing to the adverse chronological aging and health span. So I wonder if you could comment on this biological question is, if we could delay menopause, would we improve the health span? Or is there an independent pathway that we should be thinking about to improve the health span?
Can I just say one quick thing? Thank you for that question. That's really, I didn't plant that question. Just FYI. I think, so this is a real point of confusion, and partly just magnified by the press, is that the goal is not to delay menopause, right? Menopause is really defined by the cessation of menstruation. The goal is not to prolong periods for a woman's entire life. Nobody wants that. And I think it would actually be really hard. Like challenging, that would be a challenging thing to achieve. The important piece of information here is that ovaries have 2 roles, right? They're important and required for reproduction, but they also have this endocrine signaling function, right? This talking to every tissue in the body. And what they're doing, we don't understand those individual axes of communication very well. Like that's part of the research that needs to happen. But what we can say is what happens when you take them away. And in that case, right, we're talking about basically they're promoting health. And so the goal would be to preserve the endocrine function of ovaries in order to preserve health span. So we don't want to talk about ending menopause. Now, do you want to answer the rest of that?
Yeah, so very interesting question. So at least in preclinical models, if you replace old ovary from old animals with a young ovary from young animal, it's enough to extend the remaining lifespan and make them healthier, improving insulin sensitivity, reducing inflammation. So clearly, young ovary has what we call geroprotective effects. And we want to figure out what these are.
And you don't need the follicles. That benefit
That's
happens in
right.
the absence
Yeah.
of follicles.
So I agree with you that there's a hormonal influence, right? Talk to all different parts of the body, but there's more to it. Because, so for example, we have seen the signature of cellular senescence, which is like a zombie cell excreting like pro-inflammatory cytokines all over the body. You see the screaming signature in the ovary too, already such a young age. So how do we block that? I think that's going to be a very important question to pursue, especially now we know how to selectively remove or dampen senescent cells.
Yeah.
Sure.
Hi. Hi, Genevieve Neal Perry, University of North Carolina, Chapel Hill. Thank you for those talks. They were fantastic. I was a little bit more intrigued being familiar with the rapamycin and ovarian aging. I had a couple questions for you, and just wanted to say with those transplant studies, although there was some rescue, it was not prolonged, and there was some neuroendocrine dysfunction in that model. So just want to say that. But with regard to ovarian aging, I'm not sure that I totally understand what you mean by that. And because when I think about ovarian aging, I certainly think about follicular depletion. And with rapamycin, what it does, and what at least some of the preclinical data suggests, is that it kind of arrests follicular development. In addition to loss of follicular exhaustion, you also have abnormalities in oocyte quality. And one of the things that I'm worried about as a reproductive endocrinologist, people are going to think that this is also going to extend my fertility. So I would love to hear kind of what do you mean by ovarian aging?
Yeah. Yeah,
And,
thank
and,
you.
and what impact are you kind
Yeah,
of
yeah, yeah.
hoping to achieve?
Yeah. I think that's a very important point that we need to clarify. So as Jennifer said, menopause is really, in a sense, consequence of ovarian aging. Ovarian aging starts 15 years before you hit menopause, right? So if you look at, for example, reproductive function, I mean, that rapidly decline in your middle 30s onwards. And that's exactly what we see. So when we look at this, what happens during this early aging in the ovary, across all cell types in the ovary, that we see this screaming signature of mTOR activation. So in other words, there's nothing good about mTOR activation. So when it comes to ovarian follicle growth and maturation, I know it's important. So the key question is, how do we modulate to the right level? So I think it's not really, switch on and off. It's more like dimmer switch. You want to find optimal level that benefits you. I mean, if you block mTOR activation, of course, there's no follicle release. So we don't want that. So that's what we are trying to do, right? So how do we find the dampen this prematurely activated mTOR signaling signature across all cell types in the ovary? So that's what I mean by delaying or dampening aging in the ovary.
Okay, thank you.
Thank you. I love your question.
Okay. Thank you so much. That was really informative and fun. And thank you all for listening.